named as research methodology, and will be having some additional context to this research methodology. Like when we talk about the syllabus of research methodology, it constitute of the research, different types of the research, then the methods of data collection, then medical research, then biostatistics, then they have covered some aspects of animal experimentation, CPCSA guidelines, and finally declaration of Helsinki. So when you look toward the syllabus of research methodology, you can have a clear vision that it not only constitute with the research protocol to be used, the statistics to be used in the M.Pharm thesis or dissertation, but also they have touched very much to the medical research and even the use of animal experimentation during the course of your PG dissertation, right? But as it is an internal subject and we are NB accredited, NAC accredited institute, I'm taking this freedom to add some point from my side only. And apart from this research methodology, we'll be having some specialized components like how to write the research paper. Before that, how to write the review articles, how to read the research papers, and then how to identify the different journals so that your review, your research article would be published in high impact journals and in a minimum time of a period. So by the time as we came across, we'll be not only dealing with the research methodology, but also dealing with some developmental issues of the student, like we'll be having a journal club. So here you will be dealing with one presentation on already published paper, and it would be attended by all HODs, all guides. It would be a forum for the discussion, platform for the interaction of scientific data. Then ultimately we'll be having some online conferences also, so that you will be having a better exposure and your science would be contributed, not only contributed to the pharmaceutical industry or pharmaceutical academics, but also largely to the development of its to industrial application. So to begin with my presentation, I'm sharing with my screen. So I have limited or confined my presentation to roadmap of institutional research in the pharmaceutical science, and I will be giving more focus on PG research. Most of the things I will be discussing, delivering today would be also applicable for the PhD research, but I have confined myself. I will be talking more on the PG research, the research which is carried out by the student of pharmaceutical sciences at their PG level. So this is the outline of my presentation. First three slides I have dedicated to the introduction of the research. So we'll understand what is research as a meaning, as by definition. Then we'll see what you understand by the PG dissertation or PG thesis. And then we'll also touch on what are the different characters which are need to be shown by the PG student belonging to the pharmaceutical sciences. Then I will be dealing with the different research processes which are involved in the post-graduation of pharmaceutical sciences. Then we'll see how to collect the review of literature, how to gather the information from different domains which are available online, and how to jump on one specific topic, specific component, so that it will be applicable for the exploration of the research in the PG level. And finally, as it is explained by the Umekar sir, that your synopsis date submission is due in the August. So we'll see what are the different processes to write down the effective PG synopsis at the end of my presentation. So this is overall outline of the today's presentation. And I must mention in the beginning only that I will be confined very much to the research processes. Some part of it would be covered today and the remaining part would be covered in the subsequent lectures. So what is research? As defined, as it is available on the Wikipedia, The research is creative and systematic work undertaken to increase the stock of the knowledge, including knowledge of the humans, cultures, and society, and the use of this stock of knowledge to devise new application. As you can see on your screen, I have mentioned some words in the red. So it involves creativity, involve the application of systematic plans, systematic ways, and ultimately leading to increase in the knowledge. This knowledge may be applicable to human beings. This knowledge may be applicable to culture. This knowledge may be applicable to society. And when you bring all these things together, ultimately it should lead to new application. So new application may be in the form of publication, maybe in the form of patent, or maybe in the form of books or book chapter. So ultimately what it leads to your intellectual property. Ultimately may be considered as a development in the science and technology. So roughly it involves the collection, organization, and analysis of information to increase our understanding of topic or issue. So it is a very systematic process which involved collection of the data, collection of the methods, appropriate methods to be used in particular research, its organization, its result orientation, and ultimately analysis of that information, analysis of the data which is obtained during the processes of the gathering information, and leading of this to understanding of the topic or issue. So ultimately everything is leading to the depth increase in the knowledge. So at general level, research has three components. First and most important thing, to find out the question. In other words, to find out the problem. The problem may be related to society, may be related to health, may be related to pharmaceutical industry or any other issue. So to pose a question related to that problem, then you have to frame the methodology for the collection of the data to answer the question. And finally, presentation of the data to answer the question. So first, to pose the question, to gather the information related to that question and present in particular the answer to that particular question. So this is everything which begins from one problem, the methodology adopted by you to solve the issues related with that problem, and ultimately inference of that solution. And if your inference is leading to the increase in the knowledge and depth of the science, it would be considered as a research protocol or research by all means. So everything we are doing this for the thesis or dissertation, which is a essential component from administrative point of view. So thesis or dissertation is a document submitted in support of a candidature for an academic degree or professional qualification for author's research finding. The thesis must form. This is the particular definition of the thesis, may be applicable to PG or PhD. A thesis must form a distinctive contribution to the knowledge of subject and afford evidence of originality shown by the discovery of new facts and or by the exercise of independent critical power. So you can see in the blue color, distinctive contribution to the knowledge of subject. If your research ultimately leading to development of knowledge, maybe in the pharmaceutical sciences, maybe in the healthcare, maybe in the medical research, shown or demonstrated by the originality or newness, ultimately is associated with the discovery of new facts and without any bias, it could be considered as a dissertation or thesis for your PG program. So there may be question, there may be controversy. PG dissertation should be called as a thesis or it should be considered as a dissertation only. So as such, available online, no data would be reflecting it as a dissertation or it as a thesis. But most of the time, PhD documentation is called as a thesis and post-graduation research is referred as a dissertation. But most of the people use it in one way or in other way. So both the definitions are true as far as my knowledge is concerned. So what is our expectation as an institute of the reputation for from PG student, those who are involved in the research program taking at the institute level. These are some components which I have noted down. So divided into 5 category, I could have gathered more components, but I have limited to 5 components which I think are the essential component for the research to be carried out the PG program. So intelligence with the vision to see non-obvious observation. This is very important. I have seen many meritorious students who were the toppers at MPharm first year, but went to downstream level just because of their inability to see non-obvious observation. So you should develop that skill to see what is happening in the laboratory, how you get the observation. Even you are carrying out some batches, why one batch is different from another? So is there any something which is missing out, or is there something which is telling you a different idea? You can have the examples of Alexander Fleming. The discovery of penicillin is because of the non-obvious observation of Alexander Fleming. So it requires intelligence. We know you are all intelligent. As you have crossed the B-Pharm level, as you have crossed the M-Pharm first level, so there is no doubt on your intelligence or brilliance. I'm talking about the specific component, that whether your intelligence is associated with the ability to see non-obvious observation in the laboratory or not. If you're having it, it's good. If you're not having it, develop it. Try to develop it. Second most important is a self-motivation. Third, perseverance. Fourth, realistic approach. And fifth, scientific skill and art of presentation. See, most of the time, you are dealing with the research program which is given by your guide. Your guide would be busy all the time. Maybe possibly he would be interacting with you on the weekdays. Maybe possible he will be interacting with you every after 15 days. In such situation, your self-motivation is very, very important. For example, if you are not taking your research on your own shoulder, it would be a waste of time for you as well as for the institute as well as for the supervisor. So after finalization of the topic, it is very important to show the self-motivation for the planning of your program, execution of the program, and carrying out the interference of your research protocols which are actually carried out at the laboratory. Perseverance, consistency, determination is very important. Many times you will be getting frustrated because of non-availability of chemicals, non-availability of animals, non-availability of even a small equipments like pH meter, or failure of the sophisticated equipment. This is a very usual practice. Everywhere it is applicable. Not necessarily it is taking place only in the SKP College of Pharmacy County, even at the Institute of National Importance like NIPER, ICT Mumbai, or any other IIT. This is the same. Maybe sophisticated equipment is working in a well, in a functional, in a very good manner. Maybe at the time of your presence, it will not be working in a similar fashion. So your determination to proceed with the solution of that problem is very important. And that's why I'm telling you, determination. Don't run away from the problem. Try to solve them. Try to take a chase on the problem to ultimately get the solution. Find out the realistic approach. Everybody knows that we are going through some unusual situation because of this corona, and you might be getting some literature which will be telling you to find out the solution for this coronavirus. But try to work on the realistic problem, realistic approach, whether it is available in our laboratory or not, whether it is possible at our laboratory or not, what kind of equipment, what kind of instrument, what kind of facilities are required to carry out that particular research. You should have better understanding of this realistic approach to carry out the research at PG level. And fifth one, see, PG research not only involve the dedicated efforts carried out at the laboratory level, but also involve two more components. One, the scientific skill and the art of presentation. Scientific skill to carry out the research required at the PG level, to develop the design, to develop the methodology if they are new, execution of the protocol, and finalization of the protocols, getting of required information, but also involve the presentation. Presentation may be associated with the writing of the papers. Presentation may be associated with the conference presentation. Presentation may be associated with the biannual presentation carried out at your institute level, or may be associated with the university examination level. So these three components: laboratory work, development of the skill required for the carrying out the PG research, and the art of presentation, maybe written presentation or oral presentation. Both are equally important. So these are all expectation from the PG student, and I hope you would be having all these qualities. You need to develop them if you are not having it. Now coming to the research process, which you will be dealing with in your B.Pharm, M.Pharm final year program. These steps I have noted down are just a guideline. They are not rigid. They are just indicative and providing you the pathways for the completion of your PG program, completion of your research protocol as early as possible. So I have noted down first, and the most important is to identify the research problem in the broad area. So I have mentioned here, you need to understand what is the research problem. You would be working while defining the research problem in the broad area. So then you need to undergo some coursework. Like you will be having this research methodology, you can consider this as a coursework. So even if it is not mandatory from university point of view, internally it would be very essential to pass the examination carried out at the institute level related to sessional examination, related to your university examination at institute level. Then review of basic concepts and theory. This is very, very important. Like you must have gone through the Bloom's taxonomy at your degree level. So it begins from the knowledge, then understanding, and it end on the synthesis level or create level. So you are on the create level, you are on the synthesis level. But this create level or synthesis level would be justified only when you have a broad understanding of the basic concepts and theory related to your subject. For example, if you are working on the tolerance, you have designed your protocol, you have given the drug to the animals, you have collected the data, you have presented the data, but if somebody asks you what is the tolerance, you should be in a better position to tell them, to explain them what are the basic concepts, what are the biological basis of the tolerance. Then after having the in-depth knowledge or basic concepts and theories related to your subject, you have to comprehensively apply the methodology to collect the related review of literature, right? So this is very important and very critical. Having a critical data, having a critical review of literature is very important as well as difficult. Like you may be coming up across thousands of paper related to your research area. Maybe 100 of them you would be finding useful, but let me tell you very frankly, your thesis would be dependent not more than 7 to 8 very critically acclaimed research paper. So ultimately your whole thesis, whole PG dissertation would be related directly or indirectly to those 7 and 8 papers which you have finalized after talking with your guide. So this is very important that you have a comprehensive knowledge of the review of literature related to your subject. So after completion of review of literature on the online modes, by gathering the information through Google, through PubMed, through Medlines or any other platforms available, you would be in a position to define the research problem. This is again very important. Like you have gathered the information, you have collected the data, you have downloaded the number of papers in your computers. Then depending upon these data, depending upon these papers, it is very critical to find out one research problem. And this would be possible when you critically go through it. Then depending upon the research problem, you would be formulating the hypothesis, right? The hypothesis is the assumption to your data. It is the uh, presumption about the outcome of your research protocol. For example, if I'm expecting something new, something very unusual, something very innovative after completion of my research data, it would be considered as a hypothesis, right? So these are all interdependent phenomena connected with a very thin thread. Review of literature, comprehensive review of literature, then definition of your research problem. After definition of your research problem, formulation of hypothesis. Once you complete these 4 stages, very critically, believe me, you would be in a position to write your first review paper. If you are taking it very seriously, if you have collected the data to the date, you would be in a position, believe me, you would be in a position to write the review article, to send it for the publication, and by the time you complete your protocols of MRC, you would be in a position to get credit. Then formulation of hypothesis, you will plan a mode of execution, how the protocols are planned, how to execute the protocols, how to gather the information which is required to carry out the protocols, and then time come the delivery of your seminar, right? So you have planned everything in advance, you have planned the methodology, which methods are required to complete the data. Then it is obligatory or mandatory from institute point of view that you deliver a seminar. This delivery of the seminar is considered as a pre-synopsis seminar, where you will be having a presentation. Most of the time it would be considered as a PowerPoint presentation before the HODs, before your guide, in the availability of the senior professors and the principal of the institute. And there would be some suggestions, there would be modifications. Take it positively. Modify your data, the information, you can modify the methodology, you can modify the title of your presentation, title of your topic. And then after finalization, in due course of the time, the suggestions which are gathered, collected in the due course of the presentation, you would be in a position to write the synopsis. So writing a synopsis is very critical again, and would be finalized by your guide. Then next steps which are involved in the research process are design and building of the experimental setup. So you will be having the complete idea that this is the my finalized experiments which I need to carry out at my laboratory, and it should be completed within the span of 7 to 8 months. So first I will be doing a next stage, the preliminary testing to check up the settings available at the laboratory, whether your equipments, whether your instruments, whether your instruments are working or not, whether you need anything extra, whether you need chemicals, whether you need animals. So everything, you need to have the preliminary checkup for these experimental setups. Then once you have checked with these setups which are available in the laboratory, you would be in a position to run your experiments. So running the experiments independently as a part of your PG program ultimately require perseverance as well as self-motivation. So many times it happens that because of some emergency, you would be not in a position to available in the college, but make sure that there is no hampering of the data collection. If you're having a chronic protocols, make sure that some of your colleagues, some of your friends are actually carrying out the experiments. So after completing of your experiment, you will be in position to collect the data. and it should be collected in a very systematic manner. You cannot say ki the experiments which are carried out at the end of the protocols will be having the data in the first way. So there is a chronological orders, there is a chronology of the events which need to be followed very systematically, and you need to collect the data in those manners only. So after collection of the data, there would be presentation at the conferences, there would be thesis writing, and finally there would be the thesis submission. In between, after submission of your thesis, you need to have one research paper to your credit depending upon your research protocol. I'm very specific here that you need to have at least one research paper published in reputed journals of publishing group like Elsewhere, Nature Publishing Group, or uh, Lancet category of the journals, and it should be in extra to all your publication. For example, you may be getting a chance to write the research paper of your seniors, but your research paper would be your own. Keep in mind. So after that, there would be the thesis presentation before your examination, and there would be the final presentation at the time of your examination, which would be followed by your thesis and defense. Ultimately, this all step, like 12 step and examination, would be covering every aspect of your research protocol. So we'll see one by one. We'll discuss different issues related to these processes in the first lecture, in today's lecture, and these things from 8 to 12 I will be covering in the upcoming lectures. So first and most important thing where you can consider as a milestone of your research, beginning of your research, is to identify the research problem in a broad area of a science. So there are many factors. There are many issues which are governing factors, which are deciding factor of your research problem. So like collectively, I'm talking about all the branches from pharmaceutics, pharmaceutical chemistry, pharmacology, or quality assurance. There are some factors which can ultimately deciding your research problem. So many of us would be working on the diseases or social relevant issues for your PG program. We are working on alcoholism, we are working on neurodegenerative disorders, we are working on the CNS disorders. Some people from pharmaceutics might be working on cancer. I must share with you that we have got the research funding from AICT of a total outlay 22 lakhs. The principal investigator is Dr. Kamlesh Wadel, and he would be working on the cancer. Then you can work on scientific controversies available. One group is saying this is possible, second research group is saying this is not possible. So you can jump on, you can take that challenge to find out which are the scientific controversies which are available on the internet related to pharmaceutical sciences, related to health, related to medical research. You can explore them and you can find out the solution. Like you are turning to be from uh, side A, or you are turning on the side B, or both are correct or both are incorrect. So you can have the judgment that this theory is working or that theory is working, right? So always it need to be very critical to find out those controversies related to science, and it would be coming automatically to you when you try to gather the information into a depth. Like when you develop this understanding to find out the scientific controversy, you need to be considered as a candidate not for PG but the candidate for the PhD. Keep in mind. Then some group from the pharmaceutics may be working on new technologies. For example, like you might be knowing ki Rashmi ma'am or Jayashree ma'am working on some micro emulsions, liposomes for their PhD program. So microspheres are new areas to be work on, liposomes are new areas to be work on. They would be requiring some technologies where not only periphery, also your brain would be targeted, right? So central nervous targeting using this new technology would be the guiding factor for your PG program. Then the SUTIX people, pharmaceutical technology people will be working or love to work on this drug delivery system, right? So there are several drugs which are failure because of the delivery system. So we need to have the appropriate program to develop the appropriate drug delivery system for these problems. And ultimately you will be having some solutions which also may be useful when you are going to work in the pharmaceutical industry. Our pharmaceutical chemistry department would be working on the analysis of the drugs, new drugs, I would rather say, their combinations, their stability testing and validation. So this is particularly I am saying the area of Dr. Krishna Gupta, Dr. Atul Hemke and Professor Lohiya. So they would be working on the drug analysis, their stability testing, validation, ruggedness, robustness, uh, especially involving the most sophisticated equipment of the institute, HPLC, IR, FTIR, etc. So this would be the guiding factor for the student who are belonging to pharmaceutical chemistry. Then you may be working on the herbal medicines, mostly those who are having the ethnopharmacological relevance. Now, what is this ethnopharmacological relevance? You may be aware that we are traditionally using Ayurveda as well as homeopathy, Unani medicines as governed by Ayush from our generations, like from all these years we are using those medicines, herbal products, botanical based medicines for the cure and treatment of all our ailments, but the data is not available to prove its experimental evidences. So experimental evidences are lacking, and if you need to work on it, you need to find out whether these medicines, these herbal products are really used by the ethnic groups of India or not. So if it is being utilized, if it is being used, which ethnic groups, at what locations they are being used, and if they are used for what purposes, and if you find out for the specific purposes they are being routinely used by the uh, traditional community belonging to Indian population, you can certainly claim to carry out the investigation to explore the experimental evidences to uh, provide more experimental evidences to prove its benefits in the animals first and then to the human being. Then you may be working on synthesis of new molecules, like you may be aware that Professor Lohia is working on the synthesis of new agmatinase molecule from last year. He working on this concept. So there would be like derivative, there would be structural activity relationship, there would be requirement of the modification in the existing structure of the chemicals, which can be governing factor, deciding factor to carry out your research at the PG program. Then some group may be like interested to work on the industrial applications of the particular research. Like there may be some consultancy which is getting uh, interest, or which is coming from the pharmaceutical industry and you are trying to work on it. So the problem which is generated in the industry and you are providing solution to it, working at the laboratory of SKB College of Pharmacy, it may be one of the governing. Then, as I told you in my earlier talk with you at the time of your guide distribution, that interdisciplinary research would be carrying out from this session. Like we have tried it last year. Dr. Krishna Gupta is working on the analysis of agmatine in vitro study as well as bioanalysis of the agmatine. Dr. Vader is working on the creation synthesis of liposomes of the agmatine, and he's targeting some CNS disorders. Even same protocol is being adopted by Dr. Jayesh Taksande. Then Dr. Lohia, as I told you, he's working on the synthesis of some agmatinase derivative. Agmatinase is the enzyme which is involved in the degradation of agmatine, the molecule on which we are working from last 13-14 years. So this could be the interdisciplinary research and would be taking a shape when all the HODs all the research guide related to that particular topics would be sitting together along with their PG student and would be creating a common platform to explore those interdisciplinary research in the particular direction. So these are all the research problem where we work on uh, PG program. But traditionally we are all working on it, right? And these traditionally protocols are highly influenced by your guide or your supervisor. Like we don't have any control as there is a guide allotment and you have the specific guide to your credit. You have his expertise, his experience to be work on. Second, maybe as I told you, ki funded research project from the industry would be influence or can influence your dissertation program. For example, if you're working under Professor Wader, so ultimately his student would be working on the research project funded by AICD. Like as most of our students had work on the Alzheimer's disease or the protocols related to alcoholism, as we were having that project sponsored from SCRST, Government of India. Maybe sometime your own research interest would be creating the platform for your PG dissertation. But this is very rare and this is very important also because if you already are interested in particular problem, it is better to talk with your guide, convince him that this is the problem you want to work on and after the mutual agreement you can work on that particular problem which could be beneficial to both of you as well as guide and ultimately leading to beneficial effect on the institute publication. Then sometime, if I'm not wrong, it may be influenced by ongoing research at the Institute of National Importance. Like everything is available online today. So while collecting the data related to your subject, your research area, you may come across the publications which are published by the Institute of National Importance. Like the publications which are taking place at NIPER, Mohali, UICT, Chandigarh, or NIPER Hyderabad, NIPER Ahmedabad, or ICT Mumbai. The scientists who are working with this institute have created their huge impact on the scientific community of India. So it is a very natural tendency, natural process to get influenced by their research and ultimately we'll see whether this part, if not complete project, we can carry out at our laboratory or not. So out of this influencing factor, the most important influencing factor is your supervisor and your guide, right? So there would be question in your mind ki, like I'm interested in analysis and my guide is working on the synthesis. It may be ki some pharmacology student are interested in the formulation but they are working in the pharmacology in the animal studies. Some pharmaceutical chemistry student may be possibly interested in working on the animal studies but are under the guide who is working on the analysis. So don't get confused, don't get frustrated. Like consider SKB as one family, one platform where you can have the guidance from multiple component, multiple persons, I would say. Okay, like the people who are working in the pharmaceutical chemistry may be highly welcome in the pharmacology lab. The people who are working in pharmacology would be highly appreciated to work in the pharmaceutical chemistry. And we can have those research protocol which would be ultimately published in high impact journals, high impact factor journals. So there is need to work collectively. Rather, I would say we should work as a team, even it is related to individual PG program of your research. So this is my request to not only student, to all the guides available, to all the HODs of the institute to work simultaneously, to work collectively on one research program so that it will be beneficial for you as a faculty, you as a scientist, but also for the student as they would be getting early publication for their PG dissertation. So after finalizing this topics or problem which is traditionally we are working on, these are some recommendations from my point of view, right? As I'm working on one molecule from last 13 years and critically involved in the research. So these are my recommendation, only guidelines, not rigid one. It is not necessary you will be working on these recommendations or these guidelines, but I want to bring your attention that how scientific community across the globe are working on how they're creating a high impact, huge impact on the pharmaceutical sciences, why their publication is accepted, not only accepted, are highly cited in the highly reputed impact journals. So I have analyzed the data and I have like decided some factors which can be of your use, which can be maybe deciding factor for your PG research program. So we'll discuss one by them. Focus on endogenous molecule. This is very, very important. See, I want to share one example with you that while treatment of corona was decided, initially people thought ki it would be chloroquine. Then they thought ki it is azithromycin. Then they thought ki it would be hydroxychloroquine. Then they have talked about various antibodies which can inhibit the inflammation, which can be used in the treatment of coronavirus. But ultimately they have gained or they have got the influencing result with the dexamethasone. So what is dexamethasone? Dexamethasone is a synthetic steroid. So ultimately it is associated with the increase in the steroids which are available in the body itself, right? So when I look on the topics to be given to the student, when I have a question in mind which topics to be given to the student for their PG program, my first concern, my first initiative would be to focus on the endogenous molecule. Like there are many molecules, there are many peptides like neuropeptides, then amino acids, then enzymes which are available in the body. And in last 3 or 4 years, if you gather the information, you will find that there are more than 100 molecules which have high relevance to the drug discovery process. If you explore them, it may be associated with the drug discovery as well as development. So synthesis, targeting their release, targeting their degradation, and ultimately increasing their own level in the body would be one of the fantastic way to carry out the research at your PG program, right? So this is not only applicable to pharmacology student. Many times, like I may take example of insulin is available in the body, but there are some insulin injections which are available for the treatment of diabetes mellitus. But if you explore some typical the data available in last 3 or 4 years, you will realize that insulin is not only acting periphery to monitor the glucose metabolism. Rather, insulin receptors are present in the brain, which can play a very important role in the regulation of your cognition. It can play a very important role in the maintenance of brain homeostasis, right? So if you inject insulin from the periphery, it may not be reaching to the brain, right? So my suggestions that why not to prepare a delivery system, drug delivery system, taking your insulin from your depot to directly brain circuitry, crossing the blood-brain barrier. That would be applicable to the patient who are suffering from cognitive decline, the patient who are suffering from Alzheimer's disease, as well as to the patient who are suffering from diabetes mellitus and suffering from memory impairment, right? So this is just one example. You can work on several others like you can have the critical thinking of chronobiology of event. Chronobiology itself would be suggesting ki on particular time these chemicals are released in the body. You can work on it, you can work to increase their release, to increase their synthesis, and to prevent their degradation, so that their availability would be much more at the time when it would be required at a highest concentration. So think over it, talk with your guide, and decide the protocol so that you'll be working on the endogenous molecule, formulation of the endogenous molecule, and taking it to the particular part of the body, may it be brain, may it be heart, may it be lungs, or may it be kidney, ultimately associated with the recovery of the experimental animal first, and then you would be projecting this drug delivery system to the human use. So it would be breakthrough in your PG program. Next, I would be taking the example of repurposing of clinically available drugs. I must take example of pioglitazone, PPAR agonist, as it is widely accepted that these drugs are used in the treatment of peripheral disorders, right? They are the anti-diabetic agent used for the treatment of type 2 diabetes mellitus, usually increase the uh, availability of insulin. They are referred as a drugs which can be useful for the treatment of uh, diabetes mellitus, most importantly type 2 diabetes mellitus. But recent review of the literature suggests that they would be used in the treatment of some neurodegenerative disorders. Like, so they are taking the help of the concepts which are available scientifically, claiming that diabetes mellitus is associated with the memory impairment. And when one is on the therapy of pioglitazone or oral hypoglycemic agent, the chances of memory impairment or memory degradation is much lower in the patient who are suffering from diabetes mellitus. So why not to target the brain delivery of those agents which are given peripherally from the tradition to tradition? So these are just my suggestions. These are my just recommendations. So pioglitazone, even simvastatin or atorvastatin, which are lipid lowering agents, they degrade the lipids, they decreases the lipid levels, and ultimately used in the treatment of obesity, hyperlipidemia, but has got tremendous application in the CNS disorders. Nowadays, many laboratories, not across the globe, but also in the India, are targeting this HMJ CoA reductase inhibitors in the treatment of neurodegenerative disorders, including Parkinsonism and Alzheimer's disease. So you can have the formulation of atorvastatin, simvastatin, or their combination for the treatment of Alzheimer's disease. So rather than targeting HMJ CoA reductase, you would be targeting the receptors which are available in the brain circuitry. So what is your contribution? You would be making that drug available in the brain where it required the most in the treatment of neurodegenerative disorders. I would recommend the chemistry student to work on the bioanalysis. This is very important to publish the paper in the high impact journals, right? So we have started actually working in the bioanalysis. So maybe in the coming batch, maybe relating to your batch, we'll be having some protocols related to the bioanalysis. Then target comorbidity and other complications. So it is not only essential to target the main disease, like everybody is targeting cancer, everybody is targeting Alzheimer, everybody is targeting Parkinsonism disease. Keep in mind, these severe diseases or disorders are usually associated with high grade complications. Alzheimer is usually associated with depression. Alzheimer is usually associated with schizophrenia. So you can target these complications associated with these neurodegenerative disorders. Cancer itself is a horrible disease. It is associated with the severe anxiety, severe depression. Even there is an increase in the suicidal tendencies in the patients suffering from cancer because of the uh, not bearing of the pain associated with the cancer. So if you can do something for this kind of the patient, this category of the patient, it would be certainly the breakthrough in your PG thesis or PG dissertation. Then improvements of adverse drug reaction. This is again one more very interesting area. I would love to work on this area because most of the drug are not having that patient compliance. Most of the drug get failure in the post-marketing surveillance because of the adverse drug reaction. See to it whether you can work something to improve its uh, drug reactions, improve its seriousness of the adverse drug reaction. So this can be again associated with the use of the combination of your agent on which you are working with the anti-cancer agent. Certainly, you can work on those agents where adverse drug reactions are highly prominent. For example, uh, aminoglycosides, antibiotics, where adverse drug reactions are very prominent. The use of cancer chemotherapy, you can work on the prevention of adverse drug reactions with antineoplastic agents. I'm telling you, this would be the breakthrough in the drug discovery if you try to work on these problems. Then some issues like alcoholism, smoking, suicidal tendencies, which is very much available nowadays in the Vidarbha region, if I'm talking about. So you just try to get to the molecular mechanisms of these problems and to try to find out the solution for it. It's always better to work on these problems because like if you try to work on these problems, funding agency would be immediately claiming your funding claim, and maybe you'll be getting a funding for these problems. Then rare diseases, for example, thalassemia, hemophilia, sickle cell anemia, leishmaniasis, Huntington disease, which are not actually covered in your degree syllabus. So you can work on it. If you are in a position to offer solution for these rare diseases, keep in mind not only you would be in a position to offer uh, the publication in high impact journals, but if your solution works properly, you'll be getting international patents, and maybe monetary gain would be out of your imagination. I'm telling you this. And sometime you may work on the diagnosis of the disease. Like, as you know, everybody is struggling with the diagnosis of several disorders, like how they uh, available in the body and what could be the diagnostic criteria for those disorders. And like if in the beginning itself we could uh, carry out the diagnosis by taking the help of some biomarkers, taking the help of biochemistry. So this can be the research area for your PG program. And for pharmacology point of from pharmacology point of view, I would suggest we have worked on many animal models, right? But try to develop new animal model for any disorders. Like there are several animal models which are available in the world, which are available for the exploration of the drugs, but there are some limitations, right? So these limitations to work on, to find out the solutions for these limitations would be added factor for your PG program. So think over it. I'm again repeating it. Okay, these are just recommendations. These are just indicative guidelines and not the rigid one. You can talk with your guide. You can talk with your supervisor, and ultimately you can finalize. You can work on this recommendation, and ultimately you can finalize your protocols during the course of the time. Then second step which is involved in your research process is the review of literature. Uh, this review of literature basically involved three processes. First, to complete the coursework, as you are completing the subject research methodology, this would be considered as a completion of the coursework. Then review of basic concepts and theory. I would recommend you only refer the reference book for this basic concept and theories. Don't look on Google uh, or on PubMed or on any platform which are available online for the basic concepts. Refer books only, books chapters thoroughly. You can also refer Indian Pharmacopoeia or any pharmacopoeia available, and you can like develop your basic concept and theories. This is very very important. I'm telling you this because most of the time when you reach to your PG program, when you submit your PG thesis, many students, I found that they are not thorough with the basic concepts and theories. So it is very, very important that while going through, while carrying out your research protocol, you also work on the development of your basic concepts and theories. And my recommendation, only refer books and books chapters for this. Then comprehensive review of literature. Explore all the related scientific literature. This is very very important. For example, if anything, any chapter, any topic or any keyword given by your guide, try to find out everything related to it and restrict yourself to some of the reputed publishers. I'm taking name of those publishers. Restrict yourself to elsewhere, Nature Publishing Group, Springer, Taylor and Francis, and Bentham Science. Don't randomly select any other publishing groups, or don't randomly go for any other online material available with no source of information. This is my strict guideline. Refrain yourself from using those material which is freely available on Google or on internet, right? So I'm repeating it. Restrict your research to elsewhere, Nature, Springer, Taylor and Francis, as well as Bentham Science. Limit yourself to these publishing groups only. You can work on the review of uh, literature by exploring the review articles published as well as the research article published in these recent years. So look at the recent data available, like from day one. Like if I consider this as a day zero, look back down the line to 5 years and check out what are the advancement, what are the development which has been carried out in this particular area, how people working on it, which methodologies they are using, how they are facing the problem to carry out the experiment, which data they are collecting, how the data is available in these years. And then after collecting the data very thoroughly, referring more than 100 papers, more than 50 review articles or 50 research articles, confine yourself to very very specific 7 to 8 articles. I'm again repeatedly telling you, your PG program would be directly or indirectly circulating on these 7 to 8 research articles. And ultimately, you have to follow their methodology, you have to follow their concepts, their background for your research program, right? But you should be thorough enough. Whenever you are referring those 7 to 8 articles, I'm suggesting you to read out from the sentence 1 to last sentence of that paper. You should be thorough with all the references which are being used in those papers, and you should be thorough with the methodology they have used. You should be thorough with the results they have obtained. You should be thorough with the scientific development they have contributed to the pharmaceutical sciences. You should be thorough with the science they are contributing to the communities, right? So ultimately, when you get thorough with these 7 or 8 articles, you will be in a better position to frame your own research plan and to execute in a proper way. These are some cautious statements for you. Do not rely on other for literature survey. As I told you in my lecture in the beginning, your research work, if you're missing for day 1 or 2, can be carried out by your colleague. But don't rely on the others for your literature survey. This is your own research. This is owned by you. So take a responsibility, shoulder it. Shoulder the responsibility of your PG program completely. Try to find out research article by your own, by applying your brain and use your time well. Like most of the time it happens that even after submission of your synopsis, you waste so many days in the collection of the information related to your PG program that you not get sufficient time for the execution of the protocol. So keep in mind that you have not more than 7 or 8 months, and you have to plan your research program according to this limited frame of a time. Keep in mind that you're not you are not working with your PhD program, so there is a limitation of the time, and whenever there is a limitation of the time, you have to dedicate your time specifically to the each component of your research program. Keep in mind. So coming to the definition of the research problem, critically identify the gaps in the literature. So you have referred 100 articles, you found 50 of them are useful or related to your PG areas or research area. Then as I told you, you would be focusing more on 7 to 8 articles, but not more than, surely not more than 10 articles. After reading those 10 articles, you would be in a position to identify the gaps in the literature. Like these 10 research articles on your papers may be belonging to the same research groups or maybe belonging to the different research laboratories. Just explore them, just find out what are the gaps, and to find out the gaps in their literature, their research articles, you need to critically read out the limitations of their research articles, right? So where you will get these limitations of their research articles? Their limitations would be getting in their discussion part. Like in many discussion which are published in highly reputed journals, they have dedicated one complete paragraph to highlight the drawbacks, shortcomings, as well as limitation of that particular research. And after that limitations, after that shortcomings or gaps, they usually suggest some way out. They usually suggest some new methods. They usually suggest some new hypothesis which can be worked on for the new research protocol. You can certainly take help from that. So try to identify the gaps in the research publications, carefully read the limitations of the research articles published, and try to find out what author is suggesting for the future course of the action. So if you gather this particular information from all the 10 articles directly related to your research area, you would be in a position to frame your research problem very correctly and very accurately. Think why it has not been investigated yet. So it may be possible ki like somebody have tried it but they have not got the result. Maybe possible ki this is not feasible at all, whatever you are thinking. Like for example, if you are thinking on one protocol, it may not have that feasibility at the laboratory level. So think over it ki why it has not been investigated in any other laboratory. Ask yourself, do your research really provide solution for it? So you have working on one problem and you are suggesting some research plan. So by completion of your research plan, whether you are in a position to find out the solution for your problem or not, or these are some independent variable, like you're talking about the problem is a separate thing and you're providing the solution for something else. This should not happen. So try to ask the question whether your research really provides solution for your problem or not. And finally, before starting your protocol, before finalization of your research problem, think twice whether it is practical and realistic, right? So your problem may be very novel, very innovative, and you are suggesting a very innovative solution for it. But if it is not practical at your laboratory, if it is not really possible to be carried out at the laboratory of SKB College of Pharmacy, Kamti, you would be in a big problem. for its execution, right? So try to work on some practical issues, try to work on some realistic issue, right? Because as I told you, because there is a problem of a time, there is a limitation of the time, there is a crisis of the time. So you need to work on those problems only which has got the practical relevance and realistic in nature. Now you have identified your problem, you have thoroughly gone through the review of literature, you have read so many research articles, you have referred so many review articles, and you have came to the conclusion that this is the problem on which I want to work on. You already discussed with your guide. So it is almost finalized. Now it is time to finalize the hypothesis. Hypothesis, as I told you, it is a presumption, it is a assumption of your outcome that after completion of this, I would be in a position, or my laboratory would be in a position to have this solution for that problem. So hypothesis should be very specific, it should be very clear, it should be testable proposition of the prediction statement about the possible outcome of the research study based on the strong scientific data, right? So every word is very important, it should be very specific, it should be very clear, it should be testable, it should be pragmatic, right? And it should explain the statement, the possible outcome of the research study based on the strong scientific data. You cannot say that my hypothesis is not backed by the scientific data, so you cannot say anything. Your statement of hypothesis should be based on the strong scientific data, and that's why I'm urging you, I'm requesting you to carry out the thorough review of literature. This is very important. Unless and until you carry out the thorough review of the literature, you would not be in a position to form the strong basis for hypothesis to work on your PG program. So it should be novel and convincing. Like, first and most important thing, whenever you are sending your paper for publication, the first and most important thing, reviewer look whether it is novel and convincing. If your hypothesis is novel, if your hypothesis is justified, if it is convincing to the scientific community, chances are very high your paper would be accepted for evaluation, right? So these are very important points which need to be considered, which need to be looked on repeatedly while formulating the hypothesis for your research question. Then coming to plan a mode of execution, like you have finalized your synopsis, you have gone through review of literature, you have formulated your research question, you have identified your problem, then you have formulated your hypothesis. Now you have to find out the mechanisms by which execution of your protocol is feasible, possible. So make sure that research chemicals are available at your laboratory. If they are not available, start the process for its procurement. If you're asking for the gift sample from any pharmaceutical industry, make the move as early as possible. Talk to your guide. He will communicate to pharmaceutical industry so that by the time your protocol actually begins, the chemicals would be available in your hand. And this is the drawback I'm observing from last 10-12 years, that even after submission of your synopsis, when actually time comes to begin your protocols, you're not in a position to have the research chemical in your hand, and ultimately it delay the beginning of your protocol. So refrain yourself from all these complications. Try to find out which research chemicals are available at the laboratory, which drugs are available at the laboratory, whether your guide is having any drugs available with him, or whether any specific components which are required for your research protocols available in the storeroom, in the research laboratory, or not. Find out. If not, try the process for its procurement, right? Someone, if you're working on the cancer, then find out which cell lines would be used, where to procure these cell lines from, and whether feasible to procure it, because some highly sophisticated environmental condition would be required for its storage. Then whether they are not available at our institute, you're going to any other institute and to work and will be working on this cell line. So find out like how minimum time would be utilized for the working of your protocols if it is employing the cell lines. uh, Give samples if you're asking for your protocol from pharmaceutical industries. uh, Immediately write down the letters so that whenever you will be available in the college, your gift samples will be available at the laboratory. Then find out whether equipments are functional, whether accessories like it's very important because many times uh, your sophisticated equipments are working, but you are not having the accessories related with. So ultimately delaying your process of the research program. Animal studies, if your protocols are having animal studies, make sure that animals protocols are registered with CPCSCA. You attend the IAS meeting, your protocols need to be finalized, you need to have the modification in your protocol, but make sure that by the time you begin with your protocols, all these things uh, are cleared, all these things are available. So if you're carrying out the protocols outside the institute, make sure that what would be the availability of that particular person. If you're working with some specific scientist working in that laboratory, whether he will be available in that particular time or not. So these are all the issues which I am experiencing from my last 10 to 12 years, and I'm just guiding you on these lines. Ki sometime delay in the PG program would be taking place because we have not planned it properly. So plan it properly so that there would not be any problem in your execution of the program. So after planning, perfect planning, you have to deliver a seminar. This is mandatory requirement at SKP College of Pharmacy, CAMT, ki before finalization of your synopsis, you need to deliver a seminar. So don't take it as an obstacle. This is a challenge. Accept it. Not necessary कि it would be only applicable in your PG program, but lifelong you would be requiring these qualities. So developing a communication skill is very important. And I'm telling you, this cannot be inherited. I'm giving you examples of some of the faculty members who are having good orientation, good communication skill, like Madhura Dikshit, Manish Aglave, Rashmi Trivedi, Renuka Das, Dr. Krishna Gupta. They are all having good communication skill, but ask them, they would confirmly say it is not inherited. They have worked for this day and night, and ultimately they have improved it. So it's a continuous process, maybe taking lifelong to develop a skill which would be considered as a very high age for your communication. So communication skill, maybe written or maybe presentation, is a continuous process. So it would be very difficult. It would be not easy program. It would not be a easy solution. So you have to work on it. If you're not having that background, if you're like claiming it, okay, we are not from urban background and we are not having good academic uh, background, so English is very poor, but you cannot claim it. Like you need to have the pathway, you need to have the program which ultimately leading to the development of your communication skill. So try to work on it. Technical presentations are always difficult. Like I told you ki, presentations are maybe two types, like one which can be given without any preparation, but technical presentation belong to second category. They are always difficult, right? So you can talk on anything, like you can discuss cricket, you can discuss films, you can discuss politics, without any end. Without any end, you can talk on 30 minutes, you can talk on 1 hour on these topics. But while giving a technical presentation, you should be thorough enough with your subject. You should be enough with your scientific ideas, so that you would be in a better position to convince your audience, right? So they are always difficult and need to be critically presented. Don't consider it as an obstacle, right? Take it as a challenge. They are for your own development. Then preparation of presentation, right? So in the Pharm program, you would be delivering several presentations, right? So how a look on time, how much time is being allowed to you, whether it is 20 minutes, 30 minutes, 15 minutes. Who are your audience? Whether you are delivering a seminar before your juniors, whether you are delivering your seminar before your guides and colleagues, or whether you are delivering your seminar before principal or all HODs and all the guides of the institute. And be specific whether your presentation is at the time of examination, whether it is of a journal club, or whether it is a regular presentation. So all these factors can be like influencing factor for the preparation of your presentation. Then organize your talk and delivery. Organize your talk and delivery. So for example, if 15 minutes are allowed to you, and you talked for 10 to 12 minutes just on introduction, naturally you will not be getting sufficient time to discuss your results and methodology. So you have to organize your talks in a way that for everything, for from giving background to discuss your methodology, to discuss your results and actually scientific contribution, you get sufficient time. And do not read the things on the slides. Try to give it in the explainable manner, right? So do not try to read out the things. Try for it, practice it more often. Ultimately you will be having a good communication after finalization of your PG program. So coming to the synopsis writing, finally this is very important because you have a due time, and deadline is also given by principal sir. And I think they are in the August, like you need to submit your synopsis. So it will cover a title page. Then it will be having an introduction. You can consider it as a background or review of literature, or sometime you can distribute your introduction and review of literature as a separate component. In introduction, you can give the introduction of your research problem, and ultimately review of literature, which would be dependent upon those 7 or 8 research publication. Then you have to write down the objectives, and then plan of work in the bullet. So what are your plan, what are your planning for the uh, completion of those objectives. Then you have to provide the materials and methods. More specifically, like if you are belonging to pharmacology, then subjects need to mention whether you are using mice or rats. Then drugs, which chemicals you are using, which methods you are using, and you need to give the possible outcome, and this possible outcome should end or should begin with your hypothesis. But there should be like mentioning of this, we speculate that, or we hypothesize that this would be the possible outcome of the particular research program. And ultimately you have to give the references, either it is in the Harvard style or maybe in the Venko style. So it is not necessary to give the references in only one frame or in the one format. You can change it according to suggestions from guide or after discussion with your supervisor. So this is just a frame, but keep in mind it is a synopsis, right? So plan of work, material and methods, and possible outcomes should be written in the future tense. Many times it happens that you copy paste the material from your seniors and they're available in the past tense. So make sure that it is available in the future tense, right? So you, because you're going to carry it out, right? So this was all about the research programs, related issues which we are going to deal with in your PG program. So I would be happy to handle the questions and feedbacks if there arises out of my discussion or my delivery of the talk. And I just want to end my talk with the quote from Cardinal Newman, that which is given also in the FSK Barar, that a man would do nothing if he waited until he could do it so well that no one would find fault with what he has done. So don't chase for the perfection at the beginning. Work on it, try it more often, and ultimately I'm sure everybody would be perfect in their research as far as your PG program is concerned. So if there are any questions, I would happy to handle it. Thank you very much.